My name's Rachel Wright. I'm the Chief of Staff here at Evidentic, and it's my pleasure to welcome you to our webinar, Stable by Design, How Lyophilized Reference Drugs Are Changing Biologics Development. I'm going to give you a bird's eye view of what you can expect from our session today, and then it's my pleasure to introduce our speakers and hand it over to them to lead us through our session today. So taking a look first at what we're going to be getting into. So to kick us off, we're going to give you a really short overview of Evidentic and what we do. Of course, if you've already had a chance to peek at our website, you've seen that reference products are, oops. Welcome, Kasim. Are our value proposition, and this brings us to the second point. So the logistical challenges of frozen reference products are the second point that we're going to take a closer look at, and that's really going to dovetail into the testing that our team has been doing when it comes to lyophilized formats. So we're going to be taking a look at the design, the data, and of course the results. From there, we're going to take a look at the conclusion of the studies that we've run as a team, and also, of course, have time for your questions. If you do have any questions as we go, feel free to drop those in the chat. We'll be keeping an eye on the chat and trying to get to them as we go. But of course, as I mentioned, we're going to have room at the end of our session for any questions you might have. So before I hand it over, I'd love to just give a short introduction of our speakers today. So we are joined by Dr. Christian Schmitz, who also has an MBA. Dr. Schmitz is our founder and the CEO of Evidentic. He has a PhD in biology from York University and also an MBA from HHL Leipzig. His research focus has been on human cancer viruses, so with a focus there on HPV, and also immunodeficiency viruses, in particular HIV-2. In addition, he brings over a decade of experience gathered in the biotech industry, so at companies like Genentech, Multipharma, and Medizone Germany. Dr. Christian Schmitz is joined, of course, by Diana Schmidt. Diana Schmidt is our leading lab manager here at Evidentic. She holds an MSc in biology from VU Erlangen-Nuremberg and has also completed postgraduate work at the University Hospital Erlangen. Her focus there was on stem cell research, in particular looking at iPSC-based cellular modules. In addition, she's also an expert in pharmaceutical quality control, EU GMP compliance guidelines, and of course, regulatory standards. So I would say at this point, it's my pleasure to hand it over to Christian to give us an introduction of Evidentic.
Yeah, let me hand this over, take this over. Thanks very much. Do I have to? Well, how do I do this here? There we go. So you see the next slide, right?
Yes.
Okay, good. So welcome, everyone. Am I loud and clear? Yes, I am. Okay, so there's one more joining. So as you heard, Rachel is a native speaker. The next two speakers are not native speakers, so is there anything you don't understand? Please ask, but I try to do my best. So I think it's important to first, before we get to the actual core of the webinar, the lyophilized products, is what do we do, and how did we actually get to this business model? I think that's quite interesting because we get a lot of questions from our customers, even after they are repurchasing from us for several times. We understand all of a sudden they didn't know what they were purchasing. So I think it's good to have a common understanding of what we do. So Evidentic actually originated from a very practical need. And that was which we observed in the pharmaceutical supply chain. And the background really is we are a pharmaceutical trading company, and we saw that there was a growing interest especially from biosimilar companies, but also from analytical CROs and from other places to actually purchase reference drugs or just pharmaceutical products, but for a different purpose. So what were they actually looking for? So they needed reference material or pharmaceutical finished products. So when we talk about reference, just to get this clear, it's the drug that is first to market. That's what we then call a reference drug, and is patent protected and serves as the benchmark, basically. I wouldn't call it a gold standard, but for the time being, since it's the first to treat maybe a disease, it is maybe also the gold standard, but definitely it's the reference product that everyone else compares themselves to if they were to copy the product. So like a biosimilar manufacturer is planning to do so. So, but the existing supply chain from a wholesaler was not working for these companies that were asking for these drugs. They actually, and this is the majority of our customers, they need very small quantities. They don't need the whole product. They need just very small quantities, sometimes microgram amounts, but we sell milligram amounts. They also sometimes need very specific batches, or even specific presentations, or the same, all at the same time. So wholesalers can't really do this. Because they sell full packs, and for instance, CROs cannot access these drugs sometimes because they don't have the licenses for it. So they need this usually for preclinical and analytical work, and wholesalers usually are not serving these companies. So our key insight was we realized the same product is being used in a completely different context. So, and this was what shaped our solution at Evidentic. So we convert licensed medicines, so reference products, but not only, but also biosimilars, into small research units that we refer to as drug aliquots. So it is the concept is very similar to pharmaceutical compounding. And compounding in aliquotation is not much different, really. Compounding means you actually use it then for patients, and aliquotation, you use it only for research purposes, but not on humans anymore. So, and this is basically our business model. We provide access to original reference products in the format of usable for preclinical use in research. So to really get, I have just 2 minutes left really, but I have 2 slides. So I'll jump straight to the beginning, to the end of this slide here, and really it should show we've been in the market for 10 years. We served 1,400 around preclinical projects, and the core of our business model really is the logistics. And the logistics is one of the main challenges of this, because all the products that we obviously produce, the drug aliquots, are frozen at -70, and we have to get them to the customers with a good quality so that the customers can actually work as if it was the original drug. So the challenge is really the cold chain, to control the cold chain, and then bring the product to the customer in the best, shortest possible way, at the lowest cost possible, and in a very good quality. And which brings us then eventually to the topic of this seminar. Most of our products currently are supplied in a frozen liquid form. And as I mentioned before, this brings a very practical problem with it. We ship Many of our products, especially to long distance shipments like to the US or China, we ship them at -70 using World Courier or Marken, and these shipments are extremely expensive. So US East Coast currently is around 2,500 to 3,000 euros per shipment, sometimes even exceeding the actual value of what we ship to the customer. The risk factors in these shipments is temperature excursions during transit, the holdups in customs, and the consequences from this is in the worst instance, the protein degrades. We know that these antibodies, including ADCs, fusion proteins, are very, very stable, especially at these very high concentrations where we ship these drugs. Still, you don't want them to be left above freezing temperature for extended periods of time. So eventually, I've been thinking about this for about 2 years or so, because many of the products that we receive come as a lyophilized product in the first place. Say trastuzumab 150 is a lyophilized product. We have to resuspend because otherwise we wouldn't be able to aliquot it into smaller quantities. And now we're going over to the concept: how can we avoid all these difficult issues about long distance shipping, temperature control? And it's for me, it was very clear. Why don't we go into lyophilization and offer these products lyophilized? And the question is, where I hand over to Diana now, because she's done all the experiments, is what is about the quality of freeze drying the products that we tested? Is the quality the same as from our original products? And with this, I'd like to hand over to Diana.
So thanks, Christian. I hope, yeah, the handover is completed. Okay. Wait a moment. So, yes, I hope this works right now. Okay. Yeah, thanks again, Christian. I'm happy to have you all here, and we are really excited to show you our study we have performed, during the whole past year, and it was really a lot of effort. We are only 5 people in this company, but now we are really excited to show you our results, and I can tell you we have also been very surprised about the results. So let's go now to the heart of today's webinar, the actual study design. We tested 6 FDA and EMA approved biologics before and after lyophilization, and this was not a random selection, I can tell you. They were intentionally chosen to represent the full diversity we encounter in biopharmaceuticals. We decided for 3 monoclonal antibodies you can see here above, namely adalimumab, trastuzumab, and cetuximab. And we also went for Enrel with etanercept, a fusion protein, so it's smaller than a monoclonal. Also a cytokine, this is aldesleukin from Proleukin. This was, or we thought it would be a bit tricky because it's just a peptide, and peptides behave not like antibodies. And last but not least, we went also for an ADC, and here we chose trastuzumab emtansine, so from Kadcyla, because it was the first HER2 ADC. So, and for each of these molecules, we performed 3 different methods to determine, for example, thermal stability. Here we used nano-DSF. Also, we went for investigation on purity and aggregation. Here we used SEC-UV and DLS. Then, for sure, we wanted to know something about the bioactivity, so we ran bioassays. And for Kadcyla, we went a bit deeper, so we had a look at the DAR in intact mass spectrometry. So let's start with the thermal stability results. So really exciting, because thermal stability was fully retained after lyophilization. This is what I can tell you. So let's start with a concrete example. Here you can see the plot of cetuximab. And the method that was used here is nano differential scanning fluorometry, so short nano-DSF. And the principle is actually quite simple. So we track here the intrinsic fluorescence of a protein upon increasing the temperature. And once the protein starts to unfold, the fluorescent signal shifts. And from this, we derive two key parameters: the onset temperature and where the protein is starting to unfold, and the melting temperature, where 50% of the protein is already being unfolded. And here, higher values indicate greater conformational stability. So what do we see here on this specific example? The curves of the liquid and lyophilized formats, they are both perfect or almost perfectly overlaying. So they show perfect similarity. And what we can say is that the lyophilized cetuximab behaves quite similar in terms of thermal stability to the liquid reference. So let's have a look at a summary of all 6 biologics in a table, so it's easier to follow. So the table shows the onset temperature and the melting temperature values in direct comparison, liquid versus lyophilized formats. So what stands out immediately is that there are no differences, or at least the differences are minimal. So we typically see deviations of less than 1 degree Celsius, which is well within the analytical variations. So notably, even Proleukin, where we thought, okay, in terms of structure, it's the most complex and inherently least stable molecule in our panel, it shows no loss of stability after lyophilization, as shown here. So this was an important proof point for us, demonstrating that lyophilization works even for challenging formats. So let's continue with the next analysis. And here we address purity and particle size distribution. And this was determined by SEC-UV and DLS. So let's start with SEC-UV. Size exclusion chromatography separates the molecules by size, and this allows us to detect the potential aggregation and fragmentation very sensitively. So here's again the example of cetuximab. And what we can see that we have only one major main peak. So this shows us that there is no aggregation or fragmentation going on. And the second method we used was dynamic light scattering, short DLS. It measures the particle size and size distribution based on fluctuations in scattered light. The two parameters we obtain in the hydrodynamic radius and the PDI, the polydispersity index, which is here the key value. So it means that all values below 0.3 are considered homogeneous, so the sample is very homogeneous, and all values above 0.3, then your sample is unfortunately heterogeneous, so pointing to aggregation and fragmentation. And as you can see on the slide here for the DLS plot for cetuximab, you can see that we have a very narrow and very sharp graph, and again curves are overlaying. So this hints for really similar behavior in terms of purity and size distribution. And in the next slide, I can show you again the summary of all values for all 6 biologics in terms of purity and particle size. So look at the columns. We have a cumulant radius, the PDI, and the main peak percentage of size exclusion chromatography. And you can see for all 6 biologics, the values in both formats are closely aligned and show no signs of fragmentation or aggregation. So, but you might even see that, for example, for Proleukin, there's a slightly elevated PDI and a slightly lower SEC main peak in the lyophilized format compared to the liquid one, but the sample still falls within the homogeneous range. So here we can see really no fragmentation or aggregation going on. And as you will see in the coming slides, that the biological activity is fully preserved. So really good results, I have to say. So let's jump into the maybe most critical aspect, the biological function, as I told. So structural integrity matters, but ultimately bioactivity determines whether a reference molecule is a fit for purpose in your assay. So that's why we ran functional assays for every single of the 6 biologics. So let's start with adalimumab from Humira. So adalimumab was tested in the TNF-alpha reporter gene assay, and TNF-alpha binding activity was fully retained after lyophilization. So you can see overlapping curves in the dose response curves. So let's continue with Erbitux. So cetuximab from Erbitux. Cetuximab was characterized by an antibody-dependent cell-mediated cytotoxicity, so short ADCC induction, following EGFR binding. And again, no differences between both formats, as you can see in the dose response curves. So let's continue with Proleukin, which was of particular interest. And Proleukin was assessed in an IL-2 reporter gene assay for cytokine activity. As you can see, activity is preserved, which is of particular interest, as I told. And both curves are completely overlying, so there's no difference before and after lyophilization. So let's continue with trastuzumab from Herceptin. Trastuzumab is also tested via ADCC induction following HER2 binding and shows equivalent results in both formats. Again, all curves are overlaying again. So then we have, what have we left? It's etanercept from Enbrel. Etanercept is like adalimumab, a molecule that we can test in a TNF-alpha reporter gene assay. And as you can see, TNF-alpha binding was fully preserved after lyophilization. So last but not least, we have trastuzumab emtansine from Katsula. Here we performed an cytotoxicity assay. I see there's a typo. So for Katsula, we performed a cytotoxicity assay. And as you can see, yeah, cytotoxicity was fully preserved even after lyophilization, which is again of particular interest because ADCs can be for sure a bit tricky in terms of whether or whether not the drug conjugate is destroyed. So for this reason, we also went one step further for trastuzumab emtansine, and we just wanted to know, how about the drug-antibody ratio? So a very ADC-specific analytic. And in cooperation with Anu Delobel's group at Quality Assistant, an intact mass spectrometry was performed to assess this DAR value. So the DAR describes how many drug molecules are conjugated to an antibody on average. For Katsula, the DAR is approximately 3.5 to 4. So, and the result is quite clear. If you look at the values for the liquid and the lyophilized format, they are around 4, so which is really perfect. And this is absolutely, the difference is absolutely in the analytical range. So there's no trade-off that we can really observe. And this is a really important finding because it demonstrates that lyophilization does not interfere with the conjugation and that the ADC remains fully intact and in its structural complexity. And this is really what we have been really surprised about. So let's put it all together. So what we have shown today. So firstly, there's no meaningful difference in the structural integrity that we have shown in nano-DSF for thermal stability between both formats, liquid and lyophilized, and this across all 6 biologics. Furthermore, there's no aggregation or fragmentation detected by SEC-UV or DLS, so both formats show equivalent purity profiles. Bioactivity, most importantly, was fully preserved in lyophilized formats, so the potency was retained in all 6 biologics. The DAR value of Katsula was unchanged after lyophilization, being very critical for ADC characterization and comparability. So, and the key conclusion is that with the lyophilized formats, we are for the first time really able to provide you room temperature shipping significantly extended shelf life and simplified storage with no compromise on analytical quality. And this we are really, really happy about. So yeah, that brings us to the final part of today's webinar and perhaps the most interesting one, at least for our side, your questions. So we have set aside time to address whatever is on your mind. Please use the chat function. I think Rachel is going to unmute you all, so you can also just raise your hand or speak. And we are really looking forward to the discussion. And yeah, I say thank you, and I'm really looking forward to hear your questions.
Yes, with that, I'll go through and I see we've got our first question from Sophia Janun.
Yes, hi, thank you. Very interesting. Could you explain a little bit more about the shelf life? What have you done when it comes to kind of what's the expiry date that comes on the aliquot? How long data for kind of how long the Lyophilized product is giving these nice data that you showed? Thank you.
Yeah, thank you for your question. This is a really, really good question. And yeah, we are just always wondering why people are not asking for this more often. So the expiry date you find on the liquid aliquots is the therapeutical expiry date. So because the content of the aliquots is still medication. So we take the therapeutical expiry date and put it on the label of the aliquot, although we know that even in the frozen form, if you store the aliquot at -70, -80, even at -25 degrees, this already should extend the expiry date, the therapeutical it's not anymore a therapeutic expiry date. It's just a normal, usual expiry date you use also for other things. But this is already extended when for at least 4 years from our experience. So from the lyophilized products, we know because lyophilization of proteins exists since decades. And it has shown if the lyophilized product is stored in a way that the glass or the container where it's stored is sealed. So we provide the lyophilized products as a medication. It's a glass vial with a stopper and a metal sealing. So it's really tightly closed, and so the lyophilized product stays very dry. And if it stays very dry, the extended shelf life should be years, but for sure, we can just tell you on our experience. So we have started the whole project last year. So we have only, with our products, experience for 1 year now. We are pretty fine. It's nothing happened. But yeah, what we learned on lyophilization, so the whole process has been described since decades, and we can assume that our lyophilized products won't expire before years or decades.
Diana, let me just add to maybe Sophia was also after another thing. We haven't, but I'm not sure. So we'll see. We have not done any forced degradation assays on the lyophilized products. So that would be one of the next steps we would be trying to do with the ones that we've been lyophilizing right now. In terms of quality and in terms of what Diana has just presented right now, to be honest, I wasn't that surprised to see the results here. There's one reason for this. This is at least my rationale. So maybe you would prove me wrong. Is when these companies, whether it's Genentech or Roche or Merck or any of the original manufacturer produce these molecules, they obviously have one thing in mind: is the product has to be manufactured, fill finish. It has to get to a wholesaler, from a wholesaler to another distribution site. Sometimes it crosses borders several times. It's all in cold chain. We know that. And then eventually it ends up in a hospital or in a pharmacy or in a daycare clinic. And the products have to be extremely robust to make their way actually to the patient eventually. So I wasn't really too surprised that these molecules survive all sorts of lyophilization, for instance. And I think some products are lyophilized several times before they actually go as a drug substance to become a drug product eventually, and formulating and all this. I don't know how many steps are involved, and it probably depends on from product to product. So actually, I wasn't that surprised to see that they were stable. However, you're absolutely right. We haven't done forced degradation on this, and we don't know if we can actually show that by re-lyophilizing the products, we can extend their shelf life way beyond their expiration date that we see on the package that we buy when there is an expiration date, say of end of 26. How long can you store it at -70 or in a lyophilized format after 26? My experience is from doing this for 10 years, and this is not because I'm just saying this now for the sake of saying, in 10 years, we had not a single complaint of shipping these molecules in a liquid format to any of our customers anywhere. We had one complaint about actually there was aggregation happening. That was one very strange, but because all the same product was not aggregating from the same lot, but to this customer it actually happened. Besides this, these molecules are extremely stable, and we're still selling to this day. We're selling samples from 2018, so the expiry date is from 2018. To biosimilar manufacturers, for instance, who then try to compare batch by batch by batch by batch. So we have 10 batches of trastuzumab, for instance, and some dating back to 2018. So they do a microheterogeneity study from batch to batch, say. But maybe is that answering your question, or?
Yes, absolutely. Thank you.
Nice. I think we have another question actually
Sure.
in the chat. So Mattia, if you want to just chime in.
Hello, everyone. Thanks for this presentation, for this webinar. I got a couple of questions. I was wondering if I correctly understood. You took the lyophilized product and the liquid product, but is it after reconstitution of the lyophilized reference sample? So then you took the liquid and then you re-lyophilized the product and then you test that sample, or did you test the original sample?
No, Diana can answer this, but I can answer. But go ahead, Diana.
Okay, thank you. So yes, you're right. We tested two formats. So we call it frozen liquid one. This is the aliquot we are selling right now to all our customers. So the liquid solution of, for example, an antibody that was stored in the freezer, and then for sure it is unfrosted. This is let's say our reference, our liquid reference, and then a lyophilized product that was then reconstituted with water for injection was also tested against this liquid reference. So as the customer would also do it in the lab. Yeah. So once we ship the lyophilized product, the customer has also to use water for injection to reconstitute the pellet, and then further dilution can be done with any other buffer for your assay.
Let me maybe it was not because Mattia was, I think, asking for the right sequence here. The right sequence is, as Mattia just said, so we take the lyophilized product, in the case if it is a lyophilized product, say Katsula, for instance, so trastuzumab. We resuspend, we get it to the original concentration of what the manufacturer says, so trastuzumab 21 mg per ml, and then we freeze these aliquots away. However, they go through one freeze-thaw cycle, so that's the liquid one, and the ones that we that then go into the assay have at least gone through one freeze-thaw cycle. And what we haven't done so far, and this is exactly what Diana said, we use, when we prepare a lyophilized, or say a re-lyophilized trastuzumab or Katsula, for instance, get the original, resuspend, have it, freeze it once, and then ship it to our contractor, because we were going to do it in-house, but currently we do it with a contractor, and then get it lyophilized. Is that exactly, Diana, the way we do it? Or
Yeah.
yeah? Okay.
Because-
So we
because,
compared a fresh sample going through a freeze-thaw cycle once with a re-lyophilized sample, and these two were put into that assay.
Okay, and that's good to know, because then my next question would be, have you tried to see if there's differences regarding the lyophilized product? So if you take the original product,
The
which
very
is
fresh one.
yeah, lyophilized just once, and then
Yeah.
after the second lyophilization, to see if there's if there are differences, because then perhaps it may not be representative of what was the original product. Just a thought. Perhaps it's the same. I'm not sure.
Yeah, that's a very good question. Diana and I were discussing this before. If we were doing it the way you propose, so basically we get the original from Roche, resuspend, and take it straight out of into the assay. We couldn't do this, unfortunately, because the assays were done all here with collaborators, especially the bioassays. So we had to freeze them once anyway. So we treated them basically all the same way first. So we never taken out of basically out of a virgin vial, we've never taken any material out and put it straight into an assay. We haven't done that. It always goes through this freeze-thaw, one freeze-thaw cycle, or one freeze-thaw and then lyophilization step.
Yeah, no, I understand. It was just, perhaps a comment for you to consider, perhaps to test the original dry sample, reconstituted, and then put in the assay, and
Correct.
then re-lyophilized for the second time, and see if perhaps
If
the
there's any
aggregation
difference.
is the
Yeah.
same, or perhaps there's some slight differences.
We haven't done that. Yeah.
We have a few questions in the chat. So from Pushkar Ramesh, three questions all at once. Thank you, Pushkar. So the first question is, are all batches or lots of the same drug QC'd comprehensively in the same way?
I can answer that just in a very general way. The concept of Evidentic was born, maybe I didn't bring that across really well in the very beginning, was that I assumed from a very early day on that these drugs are extremely stable. I had no proof, and we just started off with the way we still do it today. Basically, it's pretty much the same as compounding a drug for, say, a DECA clinic, or so. There, besides Diana taking the original containers into a sterile laminar flow hood and then do the aseptic aliquotation process, we do not perform any QC testing on the samples. So they really go into low bind Eppendorf tubes. They're labeled with all this specific information of that specific batch number, expiration date of the original product that we just aliquoted, and so forth. All the details so that we can actually identify it. But besides this, nothing is done to these samples besides aliquotation and then straight away freezing them down to -70 degrees. Maybe that answers your question, but I'm not sure.
I think
No,
further.
sorry, quick follow-up. So I meant for the lyophilized drug, not for the original liquid format.
For the lyophilized ones. Well,
Yeah.
we've done it currently only for the 6 that you saw.
No, what I mean is, for example, if you take adalimumab and we'll order one lyophilized adalimumab now, and then
Yeah.
you'll give it from this particular batch, and 3 months later it will be from a
I see,
different
I
batch
see,
and
I
different
see,
lot,
I
and will
see,
every single vial that you aliquot
I
will
see.
that be comprehensively. So that, sorry, that's what I meant.
I got
Yeah,
it. I got it.
yeah. Okay.
To be honest, most likely no. It's because it's just way too expensive for us right now. What we're going to do, not in this comprehensive way. So when you talk about CQA or critical quality attributes, we would, I think what we would do from now on would be to do SEC to at least C aggregation. But I think we would not go through the entire cycle that we've done as an example for here in this case, adalimumab or the others, where we do all the entire set of assays. But that's obviously a very valid question.
There was one other question there, Christian, that Pushkar had, and it's how does reconstituted drug perform against liquid formats in animals, for example, rodent models, from PK and aggregation standpoints?
Short answer, I don't know. Diana, do you know?
No.
There's
What we know is, maybe I can just
Sure.
give a short hint. So we have a few customers already tested our lyophilized products, and where we know that they have done also rodent models, and they gave us already feedback that everything went quite perfectly, quite similar to the liquid ones. So this is what I can tell. It's customer experience, but here, neither here in our lab nor as a CRO cooperation or whatever, we don't do any animal testing with our products. Yeah.
Okay, I mean, these drugs are used as a positive reference, right? I mean, that to be the main use case. And then, I mean, what we kind of fear is once you inject, it can immediately aggregate before it even enters circulation. That's one possibility. And the second is you're resuspending it in water or any other excipient, you can say, in this case, and it can be excreted very quickly. So just wanted to see how we can bridge the gap between the original liquid format, which we know that how it behaves, because that would have undergone a proper in vivo yeah, studies in general. And in this one, basically the excipient is completely different, if you agree.
Excipients are usually not different. So it depends also on the product. If, like, for example, Herceptin comes already as a lyo, so
Yeah.
we are reconstituting the original vial with water for injection, and then we are lyophilizing it again in smaller volumes. But, and you know, during lyophilization, water vaporizes. And, I mean, usually there
The
are no
composition
other
should remain
excipients.
the same. Yeah, the
Yeah,
composition
there are,
should remain the same.
yeah.
But yeah, the question is valid, obviously.
Yeah, sure.
We have added to some of the products, we have added additional sucrose, but even we haven't shown the data. But basically, and this is the goal, at least this was our intention, not to change the original buffer composition by adding anything to it, neither proteins nor sugar or anything. And that was, in the first instance, this was what we tried to show, whether the proteins behave different from what we can tell for the 6 that we tested. There wasn't much of a change or no change that we could observe anyway. And we have not changed the buffer composition, and I believe adding back the amount of water that was evaporated during lyophilization should bring the proteins back to their original composition and original concentration. But
Okay.
we haven't, obviously we haven't tested this. That's true.
Okay, yeah, thank you. And when it comes to the costing itself, I mean, that's one of the main selling point, right? So that would be the main difference here.
Well,
what would be the difference that we're looking at when we are getting like a 50 or 100 microliter aliquot of a liquid.
Yeah, but that's the
to,
whole point. That's it's obviously a very practical question. That's the whole point of Evidentic, is to bring the costs down. So this also sort of is a reason for us not being able to test each individual lot, because it would make the costs go crazy. So what the rule of thumb is basically, you pay around a tenth or a fifth of the original drug in one of these, compared to one of these aliquots. That's what you can see on our website as well. So we could go into details. I don't have the prices here, but we try to make the aliquots so much cheaper that they are definitely very reasonable to obtain for research purposes. Once you get to larger quantities, so we've been asked sometimes we've been asked for 100, 150, sometimes 300 milligram or so. I mean, this is, in my terms, in my olden days, this was wholesale of pharmaceuticals. But where basically Evidentic is there for the small quantities, but the diversity across, say, batches. So you can buy different batches, but rather small quantities. And then you get away with less of an investment than buying the original containers.
And maybe just to step in, maybe that's a good point, Christian, to make sure that we share with everyone, where you can reach out to our team and get answers to more practical questions like this. So just because seeing we're at time, to make sure that you all walk away from this webinar with our website, of course,
Sure.
So you can find our product selection at evidentic.com. You can also reach out to our team, so at info@evidentic.com, and ask any questions that you might have around pricing or product availability. And of course, you're free to keep in touch with us on LinkedIn. Maybe turning it back over to you, Christian and Diana, for any last thoughts before we close the webinar.
Sure. Diana, go ahead. If there's anything you...
So are there any other questions? Just maybe if there are some thoughts on your side. So if not, what I also wanted to say is that we are always keen on broaden our portfolio. So if there's anything in your mind, we have, for example, thought about including mRNA vaccines into our portfolio. So maybe just let us know if it's of particular interest. Anything that comes to your mind, just let us know, and we would be happy to support you on this idea. So for sure, we can also broaden our licenses with our German authorities here. So we are always keen on getting information from you on what you're interested in. Yeah.
Okay. Last thing I would want to say is we're basically a pharmacy for scientists. Basically, that's how we could consider us. You can reach out to us. You can get basically every pharmaceutical product that is licensed. And just recently, for the first time, it took us about 4 months to do this. We got a number of monoclonal antibodies, including ADCs, from the Chinese market that are not available anywhere else. They're neither available in Europe nor in the US. And we just received them last week. It could be interesting because I've been to Suzhou near to Shanghai twice. I've seen this this biotech zone there next to Shanghai. It's amazing what's happening over there. They produce so many new antibodies and ADCs. So we're enriching our portfolio as well in that direction. So it's not only US, EU, but it's now being enriched by Chinese products as well. So if you have questions, just ask.
Sorry to cut you off, Kristen. I wanted to thank both you, Christian and Diana. Thank you so much for the presentation today and for leading us through the results. And of course, thank you to everyone who's joined us today. Thank you for your questions and your engagement. We've been looking forward to this for a little while now, and it was really a treat, I think, for the team to engage with you on this product that we've put so much time into testing. So say without further ado,
Thank
if you
you
have any
very much.
questions, please reach out.
Thank you.
Thank you a lot.